Hi, I'm Dr. Desirée du Sart, and I'm at the NUH Genomics Diagnostic Laboratory. My team here in the lab analyzes genomic DNA in order to identify variants that are part of a patient's clinical condition. I'm going to now take you through a workflow to show you what we do. We receive blood samples in this laboratory. The first thing we do is to extract the DNA. The next stage is to fragment that DNA and add identity markers to make sure that each patient's DNA can be identified. The next part is to put that library of DNA into a sequencer in order to generate the genetic code for each patient. This is an example of a sequencer over here. After that sequencing is performed, we will then analyze the genomic data. We take a specific part of the gene that is related to the patient's condition and we analyze it for variants in order to identify the specific variant for that particular patient. Being embedded in a hospital, I can work with the care teams and the clinical specialists who understand and know the clinical conditions. This is especially important in complex cases. The connection between the lab and the clinical team is really important to ensure that we get the right information to the right people at the right time.
Hi, I'm Elaine, a clinical pharmacist at NUH. My team works in pharmacogenomics. We use a patient's genetics profile to guide how medications are prescribed. Over 20% of patients we have tested carry variants that affect how they respond to their medications. That means the standard dose might not work or it could cause side effects. We review these results with patients upon request and build prompts in the system to inform clinicians so they understand why a medication may need to be changed. The goal is to get it right before something goes wrong.